name is Nicole. I work at Beam Therapeutics. I want to thank the organizers for inviting me to spend a little time with you and talk about what I do. This is sort of more of a philosophical talk, and it's supposed to just start a conversation for everybody to start thinking about base editing and what the possibilities of them are, instead of like a straight, flat science talk. So the human genome contains about 3 billion base pairs, and these are really just long stretches of chemical code that contains adenines, cytosines, thiamines, and these are really just the instructions for making everything in our body. So our enzymes, proteins, all the things that are the instructions for carry on life, really. And every so often there's a mistake in our DNA, and these mistakes have many times consequences. But of all the different types of mistakes that can be made in our DNA, the vast majority of them, as you can see in this pie chart in red, are actually point mutations. So it's exactly one single letter misspelling that causes some kind of disease or damage versus deletions, duplications, fusions, et cetera, et cetera. So the vast majority of diseases associated with pathogenic outcomes in humans is basically a single letter misspelling. And so what this looks like, they're called SNPs, so single nucleotide polymorphisms, is exactly one nucleobase is changed into the other, and this causes a miscoding. So an amino acid is changed from a serine to a glycine, and what this results in, for example, is a protein that's misfolded or a protein that can't carry out a chemical conversion that we're looking at. So before base editing, there's been a lot of genome editing technology that's been around, and all of them require cutting of the DNA. So we had TALENs, zinc fingers, and now CRISPR, or what we as base editors like to call CRISPR version 1.0, in which we take essentially a pair of scissors, go into our DNA at the site that we're trying to modify, cut out the bit that's not quite right, and insert something else that is hopefully going to get incorporated into the DNA and cause a repair that we would like. So CRISPR 2.0 we like to imagine more as a pencil eraser, so a more precise tool to correct a single nucleotide. And so what this looks like on a molecular level is we take advantage of the CRISPR system that can take a catalyst to a genomic site of interest very precisely using a guide RNA, open up that area that we're trying to modify, take this biocatalyst and chemically convert, for example, a cytidine into a uridine, which is then interpreted by the cell as a thiamine, and then we get a reversion at that site from a C to U to T, C to U to T. So current strategies for trying to treat genetic diseases mostly focus on the symptoms. There are some cases in which gene therapy is an option, so instead of actually correcting the DNA, they can provide some replacement protein, for example, but your DNA is still not corrected. Or in a lot of cases, genetic diseases are simply treated by trying to ameliorate some of the symptoms of the disease. But what if instead we could actually correct the DNA? We could cure, in a sense, the DNA by using a very precise tool to revert what is causing the genetic disease in the first place and not require stays at hospitals and needing to treat the symptoms. We just simply cure the genetic disease. So just to remind everyone, there's tons of pretty well-known diseases which are caused by a single point mutation. Sickle cell, and this is not an exhaustive list by any means. So sickle cell anemia, beta thalassemia, cystic fibrosis, Parkinson's disease, Alzheimer's, lots of forms of cancer, and the list goes on. And if the goal is to simply correct a single letter misspelling, a base editor could be a superior form of the technology to use. So I just wanted to take a step back for a second because my path to actually going into base editing isn't a straight one. I just would like to recognize my father and grandfather for really instilling a love of science in me, and used to do a lot of things, out in the backyard and playing with, doing the typical things like making volcanoes and things like that. That led me to pursue science in my college days, And I was actually really interested in antibiotics and natural products. And from that time in my undergraduate lab, I went on to Johns Hopkins to do mechanistic enzymology to figure out how beta-lactam antibiotics work. So my PhD in background is actually a natural product chemist. That led me to work in Professor David Liu's lab. And while I was actually working on an antibiotic project, I got distracted by genome editing and started working on building new technology. So I consider myself a natural product chemist who's been distracted by gene editing. And that work, building the technology and really being creative and making tools to be used in medicine really sort of brought me back to my parents and my grandparents in sort of the spirit of being creative and making tools to help others. And that's sort of why I went into science to begin with. So I feel like my journey has come a little bit full circle. And so when I joined the base editing project, there was a C base editor that had already been made. But unfortunately, the C base editor only could correct about 14% of all the SNPs if all the challenges of base editing had been resolved. But it turned out there was a disproportionately large amount of diseases that could benefit from what is called an A base editor because of spontaneous cytosine deamination in our cells. However, we couldn't actually make an adenine base editor by simply taking an enzyme that is found in nature and fusing it to Cas9 in the normal way that we had been working with the original base editor, because there's no known enzyme that deaminates adenines in DNA. So this is a huge problem. We really needed to create something de novo in order to enable the engineering, to enable the science, to enable the medicine. And so that was a pretty long shot. And I think I became really inspired by the possibility of creating a new enzyme because it challenged me to be really creative, and I'm a very curious person, and I really was driven by the curiosity of trying to figure out how to make something from nothing that then could be engineered to do something great. And because the need was so great and the challenge was so high, I really felt very focused in trying to make this brand new technology. And so I tried the easy thing first, as most people try, and I think is a reasonable thing to do. I found a catalog of enzymes that I thought could be fused to Cas9, and perhaps because the effective concentration of the substrate was positioned well with Cas9, I could force these enzymes to do something they normally wouldn't do. It turns out that even though you can look at tons of enzymes that can deaminate adenosine in, say, RNA or on single nuclear bases with sugars attached, they actually won't accept the single-stranded DNA as a substrate. And I think a lot of folks probably tried this approach and found out that this doesn't work and stopped, because the next sort of avenue was to create something new. And a lot of work had to go into creating such an editor. And what I wound up doing is creating a platform using the technology that had already existed to validate that one could be identified if properly pushed through a selection system. And then not only do that in what was a bacterial system, that editor could then be translated into mammalian cells and used. So what I'm showing here is just the beginning schematic of using a bacterial system to identify evolutionary products from a selection, and then converting that to a mammalian context and surveying them for adenine deamination or base editing. So what we're looking for is the bars basically to go up. So before any kind of evolution, I couldn't get base editing to work on adenine, and after several rounds of evolution, you start to see that these red and orange bars are now going up. So after several rounds of evolution, we identified quite a few mutations in a target enzyme that now allowed us to cleanly and precisely edit adenines in the context of DNA, so converting an adenine to a guanine through an inosine intermediate. And we looked at all sorts of areas in the genome, sort of very randomly, and with a very high overall editing efficiency, we were confident that this A-base editor that we had access was truly something that we might be able to push forward as a therapy. And so what's next? After you develop a technology, the hope is always to use it for a disease context. So in the case of the C-base editor, one of the targets we looked at was to change an amino acid involved in ApoE4, so that has implications for Alzheimer's disease. And what we wanted to do was revert an arginine to a cysteine, so taking ApoE4 to ApoE3. So this has implications for risk factors in Alzheimer's. And when we use the original base editor, you can see that you get C to T editing at the targeted site. So you get an amino acid reversion very cleanly with very low indel formation. So the CRISPR 1.0 versions of gene editing, again, cuts DNA, which is very dangerous for the cell. So cutting of DNA is a very bad DNA lesion. Base editing does not go through a scissor mechanism, if you will. So with the A-base editor that I developed, I was looking at hereditary hemochromatosis. So this is a liver disease, and it basically results in high levels of iron in your blood. And it turns out there's a cytosine to tyrosine mutation. So what I did is I took the A-base editor and used patient-derived cells and proved to myself that I could make a reversion precisely at the site of the disease. So I tried to change the tyrosine back to a cysteine, and I was successful in that effort. Since that time, base editors have been used in other labs across the globe in many different organisms, frogs, zebrafish, rice, all sorts of plants. I actually can't keep track anymore, but the point is more that it's not like this technology was just isolated to one lab. Once it was disseminated in the community, it was widely accepted and used in all sorts of contexts, which was really exciting and further expanded the possibility of using this base editor as a really transformative technology. So what about transforming from academics to industry? What are the major hurdles of taking something from the glossy pages of literature into patients, into real life? And it's actually a pretty big jump, as you can imagine, and one of the main reasons that I decided to pursue a career in the biotech industry as opposed to remaining in academics, which I had geared myself up for so long, truthfully. So there's three main challenges to transforming this technology from academics to a real-life setting. We need to focus more on substrate specificity. So there are on-target edits that we want to see, but there also can be off-target edits, and that's due to just the nuances of using Cas9 and having a deaminase expressed in the host cell. Delivery, I have this as a nice big picture because that's really the big issue, not only for base editing, but also for CRISPR 1.0 technologies. We have to find a way to get this big molecular machine, from your pipettes in a laboratory setting into people, and that's really, really difficult because it's large, it's charged, it has RNA, it interacts with DNA. You need to express, in some cases, in vitro protein to deliver RNP. So there's a lot of technical challenges to actually deliver this material into cells and product selectivity. And so at Beam, what we're trying to do is just that. We're working on ways to deliver the base editing technology. We're using all sorts of modalities to achieve this. We're not just fixated on one way to get into the cells. We're really taking broad strokes and trying to get this technology into people. And of course, the goal is to correct DNA in people. And we would like to take folks with a genetic disease, treat them with a base editor, and cleanly and precisely make these base editing change a protein that is no longer functioning into a functional protein, for example. And there's other possibilities for using base editing. It's not just sort of focused on one gene, one edit. technologies. You can imagine, of course, gene correction, so correction of pathogenic mutations. You can also imagine insertion of clinically beneficial mutations for upregulation or downregulation of things, suppressor mutations, things like that. Editing genetic regulatory elements, changing protein function, installing a stop codon before proteins if there's something wrong with that enzyme, and just completely abolishing its translation would make a difference. So there's all sorts of possibilities for using this technology moving forward, and we're certainly trying to push these ideas in a clinical sense at Beam. And so the pie in the sky, sort of very optimistic hope, dream for this technology is that, if somebody comes and they present symptoms of a genetic disease, they go to a hospital or their doctor, their DNA gets sequenced, a genetic disease is identified, and then we can go and find a particular base editor that will be very precise for the mutation that they're trying to revert, treat the patient. Their DNA would be base edited, DNA would be, again, sequenced to verify that the desired gene edit had been made, and then there's no further treatment needed. They are, in a sense, cured. That's it. You're just correcting the DNA, which is the problem to begin with. So that's all I have for you today. I just wanted to leave you with a thought that the hope is, to cure some genetic diseases, and that, the fear of having a diagnosis of a genetic disease is no longer there, because there is opportunity for correction to be had, and hopefully through base editor technology.